Hello everyone. Today we're going to talk about cell division. We're going to talk about two types of cell division, mitosis and meiosis. The two types of cell division that we humans can go through is mitosis and meiosis. What is the difference between mitosis and meiosis? One of them is needed to produce gametes, and that would be meiosis. So we use it so that we can make our eggs and sperm. And mitosis is needed for our cells to clone themselves, to grow, to repair any injuries. So let's say that you've cut yourself and your skin cells want to go ahead and repair the damage. So your skin cells would duplicate themselves. They would actually clone themselves. So all the cells that would come out would be exactly identical to the original skin cells that you had, and they would do that through mitosis. With meiosis, when we produce our gametes, which are our eggs and our sperm, those are not going to be identical to the parent cells. And we're going to take a look at why those eggs and sperm, they're not going to be identical to the parents. So one thing to note is that human organisms have a total of 46 chromosomes. Or we say 23 pairs, 23 from your mom and 23 from your dad. When we talk about having the full set of chromosomes, all 46 chromosomes, we refer to that as diploid or 2N. 2N meaning that you have two copies for each chromosome. And again, why do you have two copies for each chromosome? Because one is coming from your mom and one is coming from your dad. So when we want to produce our eggs and our sperm, we have to go through meiosis. And the reason why we have to go through meiosis is because meiosis is going to result in haploid cells. So these eggs and sperm, they're going to be haploid, meaning they're only going to have half the number of chromosomes. So the mom is only going to be passing on half her chromosomes in the eggs, and the dad is going to be passing on half his chromosomes in the sperm. Why is it important for the egg and the sperm only to carry half the number of the chromosomes? Because if eventually fertilization is to take place, what happens during fertilization is the DNA from the sperm is going to mix in with the DNA from the eggs. So those 23 chromosomes are going to mix in with the 23 chromosomes of the egg, and you would have a zygote that is diploid and carrying 46 chromosomes. That zygote is eventually going to grow, and it's going to start cloning itself and duplicating and multiplying itself, so that one zygote is going to turn into 2 cells, 4 cells, 8 cells, etc. So basically it's going to be going through mitosis so that it can help us, it can help that baby grow and eventually, become a full human organism. So that's, again, that's possible because it's going to be going through mitosis. So as you can see, we can go through both mitosis and meiosis. Mitosis, your cells are going to look exactly the same. Nothing is going to be different about them. With meiosis, the cells are going to be different because you're only carrying half the number of chromosomes in the eggs and the sperm. When we talk about the full set of chromosomes, we say that our cells are diploid or 2N. When your cells are only carrying half the number of chromosomes, kind of like in the case of an egg or a sperm, we call those cells haploid, or we refer to it as N. Now, for our cells to go through mitosis or meiosis, we have to go through the cell cycle. The cell cycle is divided into the following stages. We've got the G1, or we can refer to it as gap 1. We've got the S stage, or we can call it the synthesis stage. We've got G2, which is gap 2 stage, and then we've got the M. And the M could either be mitosis or could be meiosis. So Either if you want to go through mitosis, you want to clone your cells, or if you want to go through meiosis, where you want to produce your gametes, you still have to go through G1, S, and G2. And those three stages collectively, we refer to them as interphase. Interphase actually takes most of the time. So if we say the whole cell cycle, if we say it's 100%, then interphase is going to be taking 90% of the time, and mitosis or meiosis is going to only take 10% of the time. And during the mitosis and meiosis, that's when your cell is going to start to go ahead and split and divide. During interphase, your cell is not dividing, your cell is just preparing itself to divide. What does it mean, preparing itself to divide? Inside of your cells, you have DNA, you have proteins, you have organelles, you have a bunch of stuff inside of your cells. Now, before you split your cell in half, you want to actually duplicate everything inside of that cell so that when it's time for the cell to split, you would have all the contents, equal number of the contents in each cell. And that's what happens during interphase, and that's why it takes a pretty long time for interphase to take place. Especially during the S phase, that's when your chromosomes are going to duplicate. Because again, when you split your cells, you want to have all of your chromosomes, you want to have the full set of your chromosomes, especially with mitosis. And so therefore, during the S phase, your chromosomes are going to duplicate. This term right here, cytokinesis, that's when your cytoplasm of your cell is going to divide. And we will take a look at pictures of cytokinesis together. This image right here shows you how your chromosome, so this is one chromosome right here, how this chromosome right there duplicates itself. So you have a sister copy right there, and so we end up calling them sister chromatids. So these chromatids are identical to each other, and this happens during the S phase of the interphase of your cell cycle, where your chromosomes duplicate themselves. So right here, you have a duplicated chromosome. Now you still call it one chromosome, by the way, even though now you have a copy of it. Collectively, it's still considered one chromosome. So I wouldn't say that I'm looking at two chromosomes right here. I would say I'm looking at one chromosome, but that one chromosome now has a copy of itself, and this copy, we call them sister chromatids. And they are held together right here, and this region right here, we call it the centromere. Let's talk about mitosis. So again, for your cell to go through mitosis, it first has to go through the interphase. It has to go through G1, S, and G2. And as we said, during those three stages of interphase, your cell would be duplicating everything inside of it. Once it's done with interphase and it has duplicated everything correctly, it would be ready to enter into the M phase. So if the M phase is mitosis, there are four stages for mitosis: prophase, metaphase, anaphase, and telophase. Or you can use the mnemonic PMAT. So those are the four stages. So if we take a look at the cell, let's say that we have a cell right here, and let's say that this cell has a set of 4 chromosomes. It's hard for us to use like a human cell with all of its 46 chromosomes as an example, because that would be too hectic. So let's say that you have a cell and the number of chromosomes it's carrying, we're going to simplify it. We're going to say that it's carrying 4 chromosomes. And let's say that this cell right here wants to duplicate itself. You want to make exact copies, exact clones of this parent cell right there. So if you want to make exact copies of this parent cell, we got to go through mitosis. Now, before we go ahead and go through mitosis, as I said, we have to go through interphase, G1, S, and G2. That's when your cell starts to duplicate everything inside of itself, including the chromosomes. So notice how here the chromosomes, we have 4 of them, and they don't have a sister chromatid, they don't have copies. Check it out right here. Each chromosome now has a sister chromatid. What does that tell you? It tells you that your chromosomes have just been duplicated during the S phase. And once, as I said, you go through G1, S, and G2, the cell is going to be ready to go ahead and split in half. So you're going to get 2 daughter cells, and those 2 daughter cells are going to be identical to one another, and they're going to be identical to the parent cell. What happens is during mitosis, those sister chromatids are going to split, and each cell is going to get 1 sister chromatid. So again, they're going to be identical to each other and identical to the parent cell. And as you can see, we didn't lose any of our chromosomes. This cell has the full set, all 4, and this cell has the full set, all 4 chromosomes. And the reason why we didn't lose any of our chromosomes is because we duplicated them during the S phase. So therefore, since we duplicated them, each one is going to get a copy, and that's why it's extremely important for the chromosomes to actually duplicate themselves before the cell divides. Now, by the way, daughter has nothing to do with gender. That's what we call them, daughter cells. So it has nothing to do with whether this is happening inside of a female's or a male's body. At the end, mitosis is exactly the same in both females and males, and that's mitosis is when your cells want to clone themselves. Okay, let's take a look at the detailed stages of mitosis. As I said, mitosis is divided into prophase, metaphase, anaphase, and telophase. But before we start those stages, as I said, the cell has to go through interphase. So it has to go through the G1, S, and G2, and that's when everything inside of the cell is going to be duplicated, including the DNA, which would duplicate during the S phase of interphase. If you take a look at the cell, you will notice that the DNA, when it's uncondensed, it kind of looks like a ball of yarn. So this big ball of yarn is basically your uncondensed chromosomes. What does uncondensed means? It means that these chromosomes are not really they're not really squeezed into the little chromosome shapes that we have seen. Those are the chromosome shapes that I'm talking about. So you can see that during interphase, they don't look like that. And so therefore, when you take a look at it under the microscope, you're not really going to see these chromosomes because they are still uncondensed, and you're just going to see, again, like a blue like a ball of yarn right here, and that would represent the DNA. It's not going to look anything distinctive. Once you start seeing, like right here, you can start seeing those condensed chromosomes, and here you can definitely see them. That would tell you that this is the prophase stage. So the prophase stage, when you're looking at it under the microscope, how can you tell that you're looking at a prophase stage and not interphase? Prophase, that's when your chromosomes have condensed into those chromosome like shaped DNA, and so under the microscope you're definitely going to see those chromosomes. Interphase, you're not going to see those chromosomes yet because the chromosomes have not condensed yet. So after we go through interphase, and during that stage the DNA is duplicating itself during the S phase, we move on to the first stage of mitosis, and that is prophase. So during prophase, several things take place. One is the chromosomes are going to start to condense. And again, meaning that those chromosomes are going to start looking like the chromosome shapes that we are used to. And right here, they basically look like this under the microscope when you are looking at them. And again, the DNA basically keeps condensing itself, and it keeps kind of shrinking into these chromosomes right here that you are seeing. So it's obvious right there under the microscope. Another thing that happens during the prophase stage is the nuclear envelope starts to break down. So right here, the DNA, as we know, it's protected inside of the nucleus, and the nucleus has an envelope, so that nuclear envelope starts to break down. And then another thing that happens during prophase is we've got those centrioles, and out of these centrioles, these fibers come out, and those are called spindle fibers. And those spindle fibers, they're going to be pretty important because they're going to start grabbing on to these chromosomes. During metaphase, those spindle fibers, they're going to help the chromosomes line up very neatly in the middle of the cell. So those spindle fibers, as I said, they grab onto the chromosomes and they help them line up neatly. And you can see under the microscope right here, you can see the chromosomes are now lined up neatly in the middle of the cell. And those green fluorescence fibers right there that you are seeing, those are representing the spindle fibers. So this is the metaphase stage. Now, what happens during anaphase is these sister chromatids that you are seeing right there, we need to go ahead and start splitting the sister chromatids apart. So what's going to happen is those spindle fibers, as you can see, they've already grabbed on to the chromosomes. Those spindle fibers, they're going to start pulling at opposite directions. So as they start to pull, what's going to happen is that these chromatids, these sister chromatids, are going to start splitting. They're going to be moving to opposite directions of the cell. So you can see right here the sister chromatids being pulled apart by the spindle fibers. That's anaphase. During telophase, you can see that so far we have one cell, right? In telophase, this one cell starts to split in half. Okay, and that's what we call cytokinesis. So cytokinesis is where the cytoplasm of the cell starts to split so that you can end up with two cells. So cytokinesis is happening during telophase. And again, that's where the cytoplasm is dividing. So you can see right here, the cell is starting to split in half. When telophase is happening in an animal cell, we call that splitting right there, or that kind of that look that you're getting right here, we call it cleavage furrow. So cleavage furrow happens in animal cells. And this is a microscope image of an animal cell going through telophase and going through cytokinesis. And as I said, this is when the cell is splitting up in half, and we call this the cleavage furrow right there. And that's where, again, the cytoplasm is dividing, and you are going to end up with two daughter cells. Now, cytokinesis and telophase happens also in plant cells. So plant cells, they do want to grow, and they do want to go through mitosis. And so when they go through mitosis, one more time, they will have to go through the interphase, and then they will go through prophase, metaphase, anaphase, and telophase. And during telophase, when they are going through cytokinesis, they're not going to have that cleavage furrow that I just showed you previously. What they're going to have is a very thin line forming, and that we call it the cell plate. That cell plate is eventually going to grow into a cell wall. So instead of the cleavage furrow in plants, they're going to develop that cell plate, which will grow into a cell wall. Okay, now let's talk about meiosis. Meiosis is when we want to produce our gametes. As I said, our gametes are eggs and sperm. So a female would be going through meiosis to produce her own eggs. A male would be going through meiosis to produce their own sperm. Now, those gametes that are going to result out of meiosis, they are not going to be identical to each other. So when a female goes through meiosis, she's going to be producing not just one egg, but multiple eggs, about 4 eggs. Those 4 eggs are not going to be identical to each other, and they're definitely not going to be identical to the parent cell within the female's body that started off the whole meiosis process. Same thing with males, the 4 sperm that they're going to be producing, they're not going to be identical to each other, and we are going to take a look at what makes them so different and how come they're not identical to each other. And another thing is that those gametes, they're only going to carry half the number of chromosomes. So the egg is going to be a haploid, and the sperm is also going to be a haploid cell carrying only half the number of chromosomes. So like mitosis, meiosis also goes through stages, and like mitosis, before a cell enters into the meiosis stages, it has to finish interphase first. So if we have a cell, and you can see the chromosomes right here, this cell, if it wants to go through meiosis, it has to first go through interphase. And as we said, during interphase, everything inside of the cell is actually being duplicated, including the DNA. So you can see that the DNA is being duplicated, and we have our sister chromatids. Meiosis is going to result in actually 2 sets of divisions. We first have meiosis 1, where the cell divides and gives you 2 daughter cells, and then we actually have to go through another set of division, and we end up with 4 daughter cells. So at the end of the day, meiosis is going to result in 4 daughter cells. Those 4 daughter cells are not identical to each other, and they are definitely not identical to the parent cell. Where is this parent cell, by the way? This parent cell is going to be in the ovary of a female and in the testes of a male. So this is where meiosis is going to be taking place within these organs, the ovaries and the testes. Now, before we get into the stages of meiosis, I want to show you this image right here, and we call it a karyotype. So a karyotype, it's basically an image of all of the chromosomes that we are carrying inside of our body. So we humans, as I said, have a total of 46 chromosomes, or you can think of it as 23 pairs. You can see that for each chromosome, we actually have 2 copies of it. And why do we have 2 copies for each chromosome? One is coming from your dad and one is coming from your mom. And this is a normal karyotype. And how do you know that nothing is wrong with this karyotype? It's because you can see that for each chromosome, you actually have 2 copies. We say that the first 22 pairs are known as autosomal pairs. The last pair, we call it the sex chromosome pair. And the last pair, the reason why it's called sex chromosome pair, because it's going to identify the gender of the individual. If the individual is carrying 2 X chromosomes, then it's a female. If they are carrying 1 X and 1 Y chromosome, then it would be a male. So this karyotype right here, as you can see, first of all, it can tell you if there is any problem with the number of the chromosomes being carried. And you can tell that just by looking at it and seeing if the individual has 2 copies of each chromosome. If you notice that they have extra copies of a certain chromosome, or if they have less copies, then that would indicate a genetic problem. And there are multiple, like, genetic problems that can happen, and we're going to talk about how is this going to take place. But for example, Down syndrome, that's when the individuals, instead of having 2 copies of chromosome 21, they actually have 3 copies of chromosome 21. There are individuals that have X, X, and Y. So they have 3 sex chromosomes instead of the 2 sex chromosomes. Now, the karyotype, as I said, you can see the 2 copies of the chromosomes, and those 2 copies of the chromosomes right here, one is from your mom and one is from your dad. We call them homologous chromosomes. So homologous chromosomes, they're not identical. Otherwise, your mom and dad would be identical individuals. So they cannot be identical because you always have one coming from your mom and one coming from your dad. So they're not identical, but they basically carry not the same information, but similar information. So for example, if chromosome number 1 is responsible for carrying your hair and your skin color genes, then chromosome 1 from your dad is also going to be carrying the hair and skin color genes. If chromosome 6 is carrying the gene that's going to determine how tall you are, then again, your chromosome 6 from your dad is also going to be carrying the gene that will determine how tall you are. But of course, we are going to have variations between the genetic information carried by your mom and the genetic information carried by your dad. But basically, that is what homologous chromosomes are. They are similar chromosomes. One is coming from your mom, one is coming from your dad. And the thing about homologous chromosomes, they always pair up as teams. So team 3 together, team 4 together. You will never have chromosome 10 from your mom standing next to chromosome 4 from your dad. It's always the similar chromosomes. They go ahead and stand next to each other. So again, chromosome 18 together, 12 together, one from mom and one from dad. You do notice that not all the chromosomes look exactly the same. They have different sizes, and that again can help us figure out which chromosome we're looking at. And they're not normally colored, so we do stain them to help us out. So the reason why you are seeing different colors, it's because those stains, they usually bind to certain bases like adenine or thymine, guanine, or cytosine. And so depending on which base they are, that stain is going to bind to, and how much of that base do you have within your DNA, you're going to get different colors. So again, maybe chromosome 1 has more adenines compared to chromosome number 3. So again, that's why we get different colors. And the centromere location is also a difference. So sometimes the centromeres are right in the middle of the chromosomes. Sometimes those centromeres are near the top. So that's again another difference that we can tell. So from a karyotype, you can tell if you have the correct number of chromosomes. You can tell the sex of the individual, whether they are male or female, but you're not really going to be able to tell whether that person is going to be tall or whether they're going to have blue eyes or brown eyes. You're not going to be able to tell that just by looking at their karyotype. So that's what a karyotype is all about. So now we are going to go ahead and take a look at the stages of meiosis. And this is just another image right here. And you can tell right here that this individual, first of all, nothing is wrong with the number of chromosomes that they are carrying. You can see that they are carrying 2 copies for each chromosome, so that looks okay. And then you can tell that this individual is a male because they are carrying 1 X and 1 Y chromosome. Again, the first 22 pairs are what we call autosomal chromosomes, and then the last pair is called the sex chromosome. Before the cell goes through meiosis, as I said, the cell has to go through interphase first. So let's say here is the parent cell, and let's say that this parent cell is carrying 2 chromosomes, just to simplify things. Let's say that it's a diploid, and the full set of chromosomes it's carrying is 2 chromosomes. And the different colors to just represent that, okay, chromosomes from your mom and chromosomes from your dad. As we said, these chromosomes, they would have to duplicate during the S phase of interphase. So as you can see, now each one has its sister chromatids. Now, once the cell goes through interphase, it will start to divide. The first division is going to result in 2 daughter cells. And the first division, that's where basically you split your homologous chromosomes apart. So as I said, when you looked at the karyotype, remember how I said the chromosomes, they stand up together as teams. So team 7 together, team 8 together. And so those homologous chromosome teams, during the first division, that's when we split them apart. You split the homologous chromosomes apart during the first division. But then we have to actually go through a second meiotic division. And the reason why we have to go through a second division, because you can see that those daughter cells, they still have the sister chromatids. So I have to go through another division to split the sister chromatids apart. And so at the end of the day, meiosis is going to result in 4 daughter cells, and those 4 daughter cells are now going to be haploid. Why are they haploid? Because each daughter cell is now carrying half the number of chromosomes. You can see that we started off with 2 chromosomes, and that gamete is now only carrying 1 chromosome. So if this is happening inside of a female, then those are going to be 4 eggs. If this is happening inside of a male's body, then those are going to be 4 sperms. And as you can see, they're not going to be identical to each other, and they're definitely not identical to the parent cell. An important thing that happens during prophase 1 of meiosis, so it happens during the first stage of cell division of meiosis, and that is crossing over, or we can refer to it as synapsis. So what is crossing over or synapsis? This is when your homologous chromosomes, they line up next to each other. So as I said, the homologous chromosomes is 1 from your mom and 1 from your dad. So if this is chromosome number 5 from your mom, it stands next to chromosome 5 from your dad. If this is chromosome 12 of your mom, it stands next to chromosome 12 of your dad. So after they go ahead and they line up next to each other, they start to actually exchange genetic material. And that is what we call crossing over. This process is completely random. We have no idea which genetic information is going to cross over. And that is one reason why at the end of meiosis, the gametes are going to look different from one another, and that's why the gametes are going to be different from the parent cell. And if you think about it, this is one reason why if you have a sibling, you don't look identical to your sibling. You might definitely have similarities, but you're not going to be identical to your sibling, and you yourself don't look identical to your mom or your dad. And one reason is the crossing over, which is a completely random process that happens during meiosis, and that is where the homologous chromosomes, they start to exchange their genetic material. Crossing over does not happen during mitosis because, as we said, in mitosis, your cells, they're just cloning themselves, so they look exactly identical to one another. They're exactly identical to the parent. So crossing over only happens during meiosis, and it only happens once during the first prophase stage of meiosis. So meiosis, as I said, we're going to go through two sets of divisions. And so it's the same stages for mitosis. So it's prophase, metaphase, anaphase, and telophase. But we're going to give them the Roman numeral 1 because we're going to go through PMAT not once, but we're going to go through it twice. And the reason why we go through it twice is because we want to split the chromosomes so that your daughter cells would only have half the number of chromosomes. So let's say that here is the parent cell. Now again, this parent cell, before it enters into prophase 1, it will have to go through interphase. So it will have to go through G1, S, and G2. And we already talked about what happens during interphase. The cell duplicates everything inside of itself, including the chromosomes. So by the time we reach prophase 1, you can already see that the chromosomes have their sister chromatids. And during prophase 1, as we said, this is where crossing over takes place. This is when your homologous chromosomes, one from your mom, one from your dad, they stand next to each other, and that's when they start exchanging their DNA material. So crossing over happens during prophase 1. The nuclear envelope starts to break down. You are starting to have your spindle fibers being produced right here. And of course, your DNA also kind of looks like these chromosomes that we've already talked about. So they've already been condensed. Then we move on to metaphase 1. Metaphase, the spindle fibers, they're going to grab onto the homologous chromosomes, and they're going to line up in the middle of the cell. And again, your homologous chromosomes, they are lined up next to each other. So they are paired up next to each other like so, and they are basically right there in the middle of the cell. During anaphase, the spindle fibers, they grab onto the homologous chromosomes and they start to pull them apart. So you end up with homologous chromosomes on one end of the cell, and you have another set of homologous chromosomes at the other pole of the cell. In telophase, the cell itself is going to split in half. So remember, we go through the cytokinesis, exactly kind of like the mitosis, and we end up with that cleavage furrow, and we end up with two daughter cells. Those two daughter cells are now containing some homologous chromosomes, but we still have to go through a second cell division because we need to split those sister chromatids apart. Those two daughter cells, by the way, they are already haploid. So they already have half the number of the chromosomes. Remember how over here in the parent cell, if we count how many chromosomes we started off with, you have 1, 2, 3, and 4. So we have 4 chromosomes. Over here, each daughter cell only has 1 and 2. So those daughter cells are already haploids, but we still have to go through another division to split the sister chromatids apart. Okay, so after we have gone through the first mitotic division and we ended up with two daughter cells, here they are, the two daughter cells, and you can see that each daughter cell is now haploid because instead of having 4 chromosomes, now each daughter cell only has 2. Again, don't count the sister chromatids as 2 separate chromosomes. This whole thing is 1. And those 2 sister chromatids is also 1 chromosome. So this daughter cell has 2 chromosomes, and this daughter cell has 2 chromosomes. But we want now to split the sister chromatids apart, and that's why we have to go through another set of meiosis division. And so we enter into prophase 2 right away. We don't repeat interphase. So you enter into prophase 2 right away. What happens during prophase 2? The nuclear envelope breaks down, and the spindle fibers are going to be formed. No crossing over. So crossing over only happens once, and that is during prophase 1. We enter into metaphase 2, and that's where the spindle fibers, they grab onto your chromosomes and they line them up in the middle of the cell. Notice how you don't have homologous pairs, and the reason why you don't have homologous pairs because you've already split your homologous pairs during meiosis 1. So now the chromosomes, they're not going to be lined up as pairs, they're going to just line up in a single line right here down the center of the cell. Anaphase 2, those spindle fibers, they grab onto the sister chromatids, and as you can see, they start pulling them apart. So they start pulling the sister chromatids apart, and then during telophase 2, the cell itself starts to split in half. So the cell itself starts to split in half, as you can see right here, and you have that cleavage furrow right here, and you will end up with 2 cells up here and 2 cells down there. So you're going to end up with 4 daughter cells. These 4 daughter cells are haploid because now they are carrying half the number of chromosomes, and those daughter cells, they're not going to be identical to each other. They're not going to be identical to the parent cell. And those 4 daughter cells could be either 4 eggs or 4 sperm. Now, we said one reason why we are unique from our siblings and from our parents is because one is crossing over. Another thing is random fertilization. Now, let's say this is happening inside of a female's body, and the female just produced 4 eggs. We have no idea which egg is going to be chosen to be fertilized. So again, that's completely random. So random fertilization. You have no idea which egg, or if it's in a male's body, you have no idea which sperm is going to be able to make it and fertilize that egg. So again, because every single time a different egg and a different sperm could be chosen, and they could be carrying different genetic information, every time the offspring will come out with carrying different genetic information. So they're not going to be identical to their siblings. Unless, of course, you have an identical twin. That's a different story. So that's basically meiosis. We go through 2 divisions, and at the end, we have 4 daughter haploid cells. This just compares mitosis and meiosis for you side by side. So let's say here are your parent cell, and you can see that those parent cells, they are diploid, so they have the full set of chromosomes, and let's say that they are carrying 4 chromosomes. During mitosis, your cell is being cloned. You're going to have to go through interphase, and that's where, again, everything inside of the cell is being duplicated, including the DNA. And then the DNA is going to chromosomes. They're going to line up down the middle of the cell, and the sister chromatids, they get split, and you end up with 2 daughter cells that are both identical. They're both diploid, like exactly the parent cell. That is mitosis. And as I said, we go through mitosis when your cells want to clone, when you want to grow, when you want to repair damaged tissue that you have in your body. Meiosis, we go through meiosis when we want to produce gametes, so when we want to produce our eggs and our sperm. So one more time, the parent cell has to go through interphase where everything is being duplicated, including the DNA. What happens is we enter into the first mitotic division, and so we go through prophase 1, metaphase 1, etc. And that's where in prophase 1, the homologous chromosomes, they line up next to each other. So you can see they get really close to each other, and that's where crossing over takes place. After crossing over takes place during prophase 1, the spindle fibers are going to help line up your homologous chromosomes down the middle of the cell. You can see here that we still have the homologous chromosomes lined up next to each other. So during the first mitotic division, when we go through anaphase 1 and telophase 1, we're going to end up with 2 daughter cells. And what did we just split? We just split the homologous chromosomes that were lined up next to each other. But then we still have to go through another set of mitotic division so that we can split the sister chromatids apart. At the end of the the second mitotic division, we end up with 4 daughter cells, and they're all haploids. They're all carrying just half the number of chromosomes. So as we said, we are pretty unique. We're not identical to our siblings. We're not identical to our parents. And we already talked about 2 reasons why we are different because of the whole crossing over and random fertilization. There's a 3rd reason, again, why every single time the female can produce different eggs and the male would be producing also different sperm carrying different genetic information. As I said, unless you have an identical twin, then you're not going to have any genetic differences between yourself and your identical twin. You're actually going to be carrying the same DNA. So, but how come, again, we are different when we compare ourselves to our siblings and to our parents? 3 reasons. One is, let's start from the bottom. One is crossing over, and we talked about it. So crossing over happens during prophase 1 of meiosis, and that's where your homologous chromosomes, they randomly exchange genetic information. So no control over it, and we have no idea which DNA is going to be exchanged every time. So every time it's completely different genetic information that could be crossed over. Another thing is random fertilization. As I said, once a female produces her eggs and the male produces their sperm, we have no idea which sperm is going to fertilize which egg. Again, this is a completely random process, and every time, let's say, a couple conceives a child, a different sperm and a different egg could be chosen, and so therefore, again, the offsprings, they're going to look different. Now, a 3rd thing that's going to also lead to variation is something known as independent assortment. What is independent assortment? Independent assortment, if you remember, we talked about how during prophase 1, your homologous chromosomes, they come next to each other so that they could do the crossing over. Now, independent assortment is basically when your homologous chromosomes end up lining up during metaphase 1. How do they line up? So right here, let's say this is metaphase 1, right? And your homologous chromosomes are lined up right here in the middle of the chromosome. Now notice how one possibility they could line up is that all the dad's chromosomes are on one side and all the mom's chromosomes are on one side. That's one possibility. But you can have a bit of they don't have to be that organized. You can have mom, dad, and mom. So actually, depending on how they line up during metaphase 1 is going to cause variation. How is that going to cause variation? Well, after you go through anaphase 1 and telophase 1 and then go through, again, the second mitotic division, let's go ahead and check out the gametes. Notice how if they line up like so, you're going to have some gametes that have more of the dad's chromosomes and some gametes that have more of the mom's chromosomes. But if they were a bit disorganized like so, after you go through the second mitotic division, notice how your gametes can actually have a bit of combination. And so you can definitely have different combinations of gametes if your chromosomes line up this way or if they line up that way. So that's what's known as independent assortment. By the way, that doesn't mean that those are all blue, that you don't have any mom's genetic information. You do, because remember, you go through crossing over. So you do have a bit of mom over here, genetic information wise, and then over here you also still have some of the dad's genetic information because of the whole crossing over. But as you can see, this is another reason why every single time a different gamete with a different genetic information could be produced, leading to the variations that we see. Now, sometimes things go wrong, and that is known as non-disjunction. And non-disjunction is basically when your chromosomes do not separate properly. And that it has to do with the spindle fibers. Remember how we said the spindle fibers are the ones responsible for pulling your chromosomes apart or for pulling your sister chromatids apart? Now, what happens is those spindle fibers, they end up tearing. And when the spindle fibers tear, the chromosomes might not split properly, and that's known as non-disjunction. And non-disjunction is going to result in the gametes having an abnormal number of chromosomes. Either you're going to end up with an extra chromosome, or you're going to end up with a gamete that's carrying less of the chromosomes needed. So, and that can lead to definitely so many disorders. Non-disjunction can either happen during meiosis 1 or it can happen during meiosis 2. Let's talk about meiosis 1 first. So here are your homologous chromosomes, and we know that during meiosis 1, your homologous chromosomes need to split apart during anaphase 1. Now, let's say that your spindle fibers, the ones that are responsible for pulling your homologous chromosomes apart, let's say that they tear. If they tear, what ends up happening is your daughter cells are now going to have, one of them is going to have extra set of chromosomes, and the other daughter cell is now going to have fewer chromosomes. And so when you take a look at the gametes that would result at the end of meiosis 1 and meiosis 2, some gametes could end up with an extra copy of a chromosome, and we call it N+1, and some gametes, now they're going to have less number of chromosomes, and we refer to that as N-1. We refer to N+1 as trisomy, and the reason why we call it tri, because now instead of having 2 copies of each chromosome, now you have 3 copies of a certain chromosome. And then here, N-1, as I said, you need 2 copies of your chromosomes. Now you have, let's say, 1 copy of that chromosome, and we call it monosomy. So mono meaning 1, and then tri again meaning 3. And monosomy or trisomy, if you have a fewer number of chromosomes, or if you have more chromosomes in your gametes, that is definitely considered abnormal. Also, non-disjunction can take place during meiosis 2. If we say that during the first set of division, everything went fine, and your homologous chromosomes, they were able to split perfectly. So nothing went wrong during the first mitotic division. But then during the second mitotic division, when you were supposed to split your sister chromatids apart, so let's say this is where things have, gone wrong, and your spindle fiber again tore, and the sister chromatids again one more time failed to separate. Same thing, you can end up with gametes that are trisomy, and you can end up with gametes that are monosomy, and you can end up actually in this case with gametes that are absolutely normal. So this is basically non-disjunction, and that is when your chromosomes are failing to separate. Either the homologous chromosomes are the ones that are failing to separate, as you can see right here, or it could be your sister chromatids, they are the ones that are failing to separate. And the reason behind it, it's your spindle fibers, they are tearing. And that could result in gametes that are carrying extra copies of chromosomes, or they are carrying a few number of the chromosomes. An example of non-disjunction that would result in the chromosomes failing to separate, and so therefore the offspring that would result gets an extra copy, so a trisomy of chromosome 21. So if you look at their karyotype, as you can see, a normal karyotype should only have 2 copies for each chromosome, one from your mom, one from your dad. But if something goes wrong, like in non-disjunction, and your homologous chromosomes or your sister chromatids failed to separate, you can end up with an extra copy of the chromosome. So in this case, this individual gets an extra copy of chromosome 21. That's what results in the disorder known as Down syndrome. Now, does it only happen with chromosome 21? And do we only get the disorder known as Down syndrome? Absolutely not. There are so many different disorders out there, and one individual could have, let's say, XXY. One individual could just have one sex chromosome, so just one X. And again, there are other disorders that can happen with the other chromosomes as well. You can either have an extra copy of it, or again, you're missing one copy. So this was our discussion about mitosis and meiosis. Again, meiosis, we only go through it when we want to make our eggs and our sperm, and it's absolutely important that our eggs and our sperm only have half the number of chromosomes. Mitosis, you go through it every time you want to repair an injury, or you want your cells to basically grow, and so your cells are going to be cloning themselves. When cells divide, whether it's a plant cell, animal cell, when they divide, they have to have a system of when should they stop their division and when should they go ahead and start their cell division. Because if they don't have a control over it, things can go out of control. And if things go out of control, this is how we end up with cancerous cells. Because cancerous cells, they are totally out of control. They divide, and they divide rapidly. They divide without stopping, and they can start invading other parts of your body. And so the cell cycle that we were talking about, the G1S and the G2, during that interphase, especially during that interphase, there are actually several check marks that needs to be checked before they can go ahead and start splitting. And there are proteins that are responsible for sending like stop signals or go ahead signals for your cell to either proceed with the cell cycle or for your cell to go ahead and freeze and stop and don't go through the cell cycle. Things can go wrong, and that cell cycle, the proteins that are being produced could become mutated, and so therefore the whole stop and go ahead signals are going to be totally out of control, and your cancer and the cells, that's when they become cancerous, where they are not responding to the stop signals, and the cells continue to divide non-stop, and they divide excessively to the point that they start invading your other tissues of your body. And so they kind of take over, and your normal cells cannot really survive with all of that accumulation of cancerous cells over them, and your actual cells can start degrading, and your organs can start like shutting down. And the cancer basically spreads throughout your body. So it's definitely an abnormal behavior, cancer cells. They are acting very abnormally, and they divide excessively, and that's because your body and your DNA has gone through some sort of mutation, some sort of genetic change. Now, what are some reasons behind these mutations? It could be several things. You could have been exposed to a lot of UV rays. So the UV rays can actually penetrate your cells and penetrate your basically your DNA and starts messing up your DNA, and that can lead to cancer, specifically skin cancer. But again, most cancer, it starts off in one region, but then it can have the ability to spread. Okay, so an individual could start off with lung cancer, but then the cancer can start spreading to other parts of their body. You can start off with skin cancer, but then it can start to spread throughout your whole entire body. And again, that's going to cause tumors. So when someone has a tumor is basically where you have, within this region of your body, your cells have started to duplicate and replicate super fast because of a mutation. Now, some of those tumors could be benign, and that's when they are not really spreading to other parts of your body and creating a bigger problem. And sometimes they are malignant, and malignant tumors, those are the ones that are going to be dividing very, very fast, and they are going to be spreading throughout your whole entire body. And again, they could start entering into your bloodstream, and they can start spreading throughout your whole entire body. And that's when, for example, they would tell the patient, okay, you have an advanced stage of cancer. An advanced stage of cancer would mean that the cancer cells are not now residing in one region of your body. They've actually spread throughout various parts of your body. And that's when the treatment gets harder. So the treatment for the cancer cells that have spread throughout your body is definitely tougher, and it will definitely take longer to treat it, and hopefully it's going to be able to get treated because there are no guarantees depending on how much damage happened to the organs. And so again, catching cancer at an early stage is definitely very, very helpful when it comes to treating it compared to realizing that someone has cancer after it has spread out. One thing we calculate is the mitotic index, and the mitotic index gives us an indication of how many of our cells, when we take a look at a tissue, how many of them are in mitotic stages, so prophase, metaphase, anaphase, and telophase. And when we compare a normal tissue to a cancerous tissue, you will actually notice a difference. You will notice that the cancerous tissue, most of the cells that are going to be in that tissue, they're going to be rapidly dividing. So when you take a look at them, you're going to find a lot of the cells are going through mitosis. So the mitotic index of those cancer cells are going to be way higher compared to calculating the mitotic index of a normal cell. And so for your lab report, you would need to take a look at patient slides and take a look at their tissues and calculate the mitotic index for normal tissues and cancerous tissues. And again, you should notice that the cancerous tissues, they're definitely going to have a higher mitotic index compared to the normal tissue. So I'm going to show you how to calculate one of them, and then you guys can do the rest on your own. So let's say that here is a tissue of a patient who has lung cancer. Now, the first thing you would want to do is figure out how many cells are in the mitotic stages. And again, what are the mitotic stages? It's prophase, metaphase, anaphase, and telophase. Interphase is not a mitotic stage. Interphase is what needs to happen before the cell enters into mitosis. So remember how we said in interphase, how can you tell if a cell is in interphase or prophase? You look at how dark the DNA is, because during interphase, the chromosomes have not been condensed yet, but in prophase, the chromosomes do start to condense, and you get those condensed chromosomes, so it's actually more visible under the microscope. So these cells right here, the ones where the DNA is not really that dark and you don't see lines, those would be interphase. these ones right here, where the DNA is much darker, and again, the reason why it's also much darker because now they're condensed, and so when we stain them, they will take in more color, so they will look more vibrant under the microscope. These would be the prophase. So what you want to do is you want to go ahead and start calculating how many are in prophase. So I see that this one right here is in a prophase stage, and that one right here, prophase. Okay, what other stages do we have in mitosis? We have metaphase. Do you see any cells in metaphase? I see one right here. So this one is in metaphase. Okay, what about cells in anaphase? I see one right there. And then telophase. I see one cell right there. No, like if you note, the cell that in telophase, it hasn't split yet, right? So it hasn't split yet, so I'm still going to count it as one cell, okay? Because it hasn't split yet. So to calculate your mitotic index, what you would want to do is basically start adding up your the stages that you've seen. So I see 2 in prophase, right? I see 1 in anaphase. I see 1 in metaphase, and I see 1 in telophase. That's 1 right there. So I see 1 in telophase. Okay, and then you divide by the total number of cells. So if we count the cells together, you should count a total of 20 cells. So 1, 2, 3. Yeah, so there are 20. Okay, again, you count the telophase as one cell because you still don't have complete separation. So now calculate it. So that would be 5, right, divided by 20. So your mitotic index is 0.25. Now, if you compare the mitotic index of this cancerous tissue with the normal lung tissue, we should expect that the mitotic index for the normal lung tissue, it should definitely be lower than 0.25.